Good afternoon, everyone. I'm Maria Malopoulos, a scientist at the Institute for Education Research at UHN, otherwise known as TIER. And on behalf of TIER, I'd like to welcome you all to the Big Ideas lecture series. I'd like to begin with an acknowledgement of the land on which we work, study, and live. We recognize that many Indigenous nations have a long-standing relationship with the territories upon which TIER is located that precede the establishment of the University Health Network. We acknowledge our presence on the traditional territory of many Indigenous nations, including the Mississaugas of the Credit, the Anishinaabeg, the Chippewa, the Haudenosaunee, and the Wendat peoples. Today, this land is home to many diverse First Nations, Inuit, and Métis peoples, and we are grateful to have the opportunity to live, work, and gather on these territories. We acknowledge that Toronto is covered by Treaty 13, signed with the Mississaugas of the Credit, and the Williams Treaty, signed with multiple Mississaugas and Chippewa bands. Since this lecture is a virtual gathering, a singular land acknowledgement does not capture the richness of our distribution across many locations. There are few places on Earth that someone before us has not called home. I encourage you to consider your own position with regard to the land where you find yourself. And if you're not sure how to locate the Indigenous territories, treaties, and languages that shape both the past and present of your current location, I invite you to take a moment now to look it up. One of the benefits of this virtual format is that we can make use of virtual resources. Take a moment to open up another browser window, type in native-land.ca. It is a crowdsourced interactive map built around principles of land as pedagogy. The intention is to use these resources as an opportunity to critically engage in discussions about colonization, land rights, language, and Indigenous history. Land acknowledgements are only a starting point for larger conversations. More concrete acts of restitution and transformation are needed to address underlying inequities and blatant discrimination in the distribution of resources between Canada's First Peoples and settlers. Before I introduce our speaker today, some housekeeping details. You can all ask questions during and after the presentation via Slido, and the link should be on your screen, and there will be time at the end for Dr. Hecker to answer questions. Also, please note that the session will be archived on the TIER website for later viewing pleasure. And if I could ask that you please fill out the evaluations for us, it's very helpful as we plan future Big Ideas lectures. It's my great pleasure now to introduce our speaker for today's session, Dr. Kent Hecker. Dr. Hecker is a professor of health professions education in the Faculty of Veterinary Medicine with a joint appointment in the Cummings School of Medicine at the University of Calgary. He's the inaugural Chief Assessment Officer for the International Council for Veterinary Assessment. Dr. Hecker studies performance in health professions education, focusing on three areas: selection of applicants, assessment of student trainee competency development across the healthcare continuum, and the application of neuroimaging methods like functional magnetic resonance imaging, fMRI, and electroencephalography, EEG, to assess learning, reasoning, and decision-making. Kent has been working at a fascinating intersection of measurement, learning, education, neuroimaging, and psychology for the past 7 years. His collaborations with neuroscientists, psychologists, clinicians, and educators have led to some of the first novice expert studies assessing the neural correlates of learning, reasoning, and clinical decision-making. He's the principal investigator of the Health Education Neuroassessment Laboratory, CENAL, a Canada Foundation for Innovation-funded lab where a multidisciplinary team combined brain imaging techniques with educational and assessment methods to explore how neuroeducation studies could inform learning, decision-making, teaching, and assessment practices in health professions education. Today's talk is titled, Why the Brain? Can Neuroimaging Inform Health Professions Education? And Dr. Hecker, I turn it over to you.
Thank you, Maria. Thanks for the great introduction. As indicated, I'm a professor both in the Faculty of Veterinary Medicine and at the Cummings School of Medicine at the University of Calgary. My training is actually in educational psychometrics, so disclaimer, I am not a neuroscientist, I am neuroscientist adjacent. Over the course of my career, I've become increasingly interested in the potential application of neurosciences as well as neuroimaging to better understand learning, teaching, decision-making, and assessment in health professions education. As indicated, I do have a conflict of interest. I am the Chief Assessment Officer with the International Council of Veterinary Assessment. So this organization is the veterinary equivalent to the Medical Council of Canada and the National Board of Medical Examiners within the United States. So let's begin. There have been calls to use neurosciences and neuroimaging in the health professions education field for decades. Here are just two of many quotes that are out there. Elstein in 1978 stated that until we are effectively able to understand neurocognitive processes underlying decision-making, early researchers in clinical reasoning remain correct in that we will not be able to dependably teach or evaluate one of the most important characteristics of being a physician. Kurzweil in 2012 was even more direct, stating, the closer we can get to being able to precisely understand the brain and decision making, the more able our tailoring to impact this area of clinical education and in turn performance of clinical diagnosticians. Indeed, as I was moving through my PhD studies, I remember conversations with my supervisor around the potential of using neuroimaging to understand brain processes as people learn and make decisions. We thought it would provide a number of answers to our questions within the field of education. I can now say, with some of the pilot research projects we have done, some of the studies that we and others have published, it has instead increased the number of questions of where, how best, and when to use these methods. So what is neuroeducation? Educational neurosciences or neuroeducation is at the confluence of an overlap of many different disciplines, primarily neurosciences, education, and psychology, with the aim, as Brolt-Foisy explains, to address three overarching areas, which include examining the biological process to support and constrain and/or constrained learning, exploring the neural correlates of academic skill, and investigating how teaching practice or educational intervention impact function and/or structure. Ultimately, what we want to use is behavioral and neural data to improve decision making and optimizing the learning environment and educational practices. So what are some of the imaging methods and equipment used in this area? So in the upper left-hand corner is a functional magnetic resonance imaging machine. While a number of studies, including two of ours, have used this methods, I won't be focusing on results from fMRI studies in this talk. In the upper right, we have an electroencephalogram, an EEG. I'll be focusing on this technology throughout this presentation. Bottom right is a mobile EEG machine called the Muse. We have run studies with this device, and I'll touch on it briefly near the end of this talk. And in the bottom left is a portable eye tracker. These devices are becoming more commonplace in medical education studies. We're using these devices coupled with EEG in my lab. So with that, it's just a bit of a background. I have only one high-level objectives for this big idea talk. Provide an example of the trajectory of how our team applied a research paradigm, learning theory, imaging method, and specific component of the human event-related potential in ERP in the context of health professions education. Interwoven in this will be a story of the serendipity of science, beginning with finding a long-lost friend and how a team of lab was built with various background and skills, as indicated before, with neurosciences, psychometrics, clinical expertise, educational expertise, just to name a few. What I'll end with are some practical challenges that need to be considered and addressed as we move forward within this area of research. In doing this research, I find myself navigating between pragmatism, paradoxes, theories, and the practical meaning and applications of what we're finding. So what I'm presenting are what I consider preliminary applied experimental designs and findings which may or may not provide suggestions for moving forward as to how best to proceed. So with that as background, what I want to present is an example of moving from basic sciences concepts into applied neuroeducation research to the possibility of incorporating those findings into classrooms or learning settings. So the idea is moving from blobs, which I'll show you in just a bit, to liver pathologies, which I'll show you near the end. As I mentioned, this story and research began with finding a long-lost friend. So in 2009, a paper was published entitled Learning to Become an Expert, Reinforcement Learning and the Acquisition of Perceptual Expertise. The author is Olaf Krugolson, who it turns out is a childhood friend who I lost contact with for 20 years. As an aside, it was actually my mom, hope you don't mind me saying this, it was actually my mom who said that she read an announcement for an award that he had won. I contacted Olaf out of the blue, and in the email, I made a few disparaging remarks about his ability to play basketball, which he and I did as kids. When I didn't hear from him, I thought I offended him. Turns out the email address I used was no longer active. When I sent it to another address, I got an immediate response. Literally, it was within minutes. Thus began a number of collaborative research projects. I asked him about applying his work in health professions education. In the paper, he talked about perceptual expertise of bird watching and dog judging. And I asked him, had he ever thought about applying this learning paradigm in health professions disciplines? Say, thinking about perceptual expertise in learning anatomy or pathology, etc. He hadn't. So at the risk of embarrassing Olive and myself, as well as my sister, here's us in the early 1970s, and again in 2017. We laughed that two salmon-armed kids are now doing science together. And by the way, I do have permission from Olive to use this picture. So let's get back to science and Olive's paper. So Olive studied whether novices could learn two different blob families, A and B blobs. I was excited about this paper for a number of reasons. The use of neuroimaging to study a reinforcement-based learning task. It reported a neural signature, an event-related potential, an ERP, called reward positivity that can track learning. Olive was able to categorize in this paper those that were able to learn the shapes and those that did not. He demonstrated a behavioral and, interestingly, a neural profile of both groups. Finally, it got me asking, could we apply this learning within the health professions program? Before going from blobs to learning anatomy, to 2D versus 3D learning, to clinical categorization tasks, to veterinary pathology, a brief introduction to those in the Zoom room, to a reinforcement-based learning task, EEG and event-related potentials, and specifically an ERP component that has been reported as a neural signature of learning, reward positivity. A disclaimer, this component is used as an exemplar. Are there others? Yes, there are. For the sake of this talk, we focus on just one component. So just to introduce you to a reinforcement-based learning task, this is just a generic example using Olive's blobs as the example. First, a participant sits in front of a screen and a fixation cross is presented for a period of time, 250 milliseconds. Second, one family of blobs are shown. Third, a label is provided, A or B. Fourth, the participant is asked to press a button for either correct or incorrect. There's a time limit. If the person does not respond, it is assumed that their response is incorrect. Fifth, as a result of the response, feedback is given, either correct or incorrect. This whole trial takes approximately 3 seconds. In Olive's paper, there were 10 blocks. Each block was 120 trials. We are interested in the neural component that is time locked to when feedback is given, as indicated by the reward positivity box. So, what are we using? We're using EEG and we're measuring electrical brain activity at the scalp level. What we are interested in are the brain activities recorded over the entirety of the paradigm and specifically the event-related potential. Shown here is Bethan Wilson in the foreground participating in one of Dr. Sarah Anderson's research projects in our lab. So what is an event-related potential? An ERP or ERPs are time-locked to events of interest. In our example, we are interested in brain activity that are time-locked to correct responses and data that are time-locked to incorrect responses. So ERPs are extracted by averaging EEG signals over timed events. This allows for EEG signal unrelated to the event to be averaged out. The data are then plotted. X-axis is time and a Y-axis represents scalp potential in microvolts, and it shows deflection patterns. The negative voltage is plotted up and positive is plotted down by convention. This can get confusing, so please refer to the Y-axis as we move forward within this talk. The ERP can be broken down into components and examined. In this study, we are targeting one specific component labeled reward positivity and its effect on N2 amplitude measured approximately between 250 to 320 milliseconds after stimulus onset. So what is reward positivity and how is it measured? First, it is a signal generated by the anterior cingulate cortex and measured over the medial frontal cortex. It is associated with internal evaluation of external feedback during learning. It has been stated that it scales like a prediction error when there is a difference between what you expected given your response and what you received by feedback. If there is a big difference, the larger the reward positivity, which is reliance on feedback, the larger the amplitude of the N2 components. Over time, as people learn, the amplitude diminishes, and that's just shown as the arrow that we're looking at amplitude of this N2 component. In other words, the onset of learning creates a diminished reward positivity amplitude, and that's what we're measuring. And really what we're interested in are the changes in this component over time. So with that, just as a little bit of a background and getting back to Olof's blob work, he first showed within his paper differences in those that learned, and he labeled them as high learners, and those that categorized as low learners over time given the reinforcement-based task. He then showed, and this is what really got us excited, or me excited when I first read his paper, was he then showed that the learning creates a decrease in the reward positivity amplitude between the beginning of the experiment and the end of the experiment. So the amplitude diminished, indicating onset and maintenance of learning in high learners he identified as indicated by this graph. What was also interesting is in the low learners, there's no decrease in reward positivity amplitude. Oops, my apologies. From those initial discussions, we were fortunate enough to embark on a few research projects applying his basic neurosciences lens in applied learning contexts. We have used fMRI, as I mentioned before, to look at novice expert differences when reading and diagnosing difficulty in easy clinical scenarios. That is work published by Herska et al. However, Olof and I kept having conversations about the use of whether we can directly relate the findings from being in a magnet to learning in a more natural environment. We kept coming back to the use of EEG and the building of his blob and building off his blob work. At University of Calgary, a colleague and anatomist, Heather Jimninsky, was interested in studying learning and curriculum. A graduate student approached her, Sarah Anderson, who was interested in exploring spatial learning and anatomy using quantitative methods. That's Sarah in the background. I suggested that why don't we try something high risk, high reward? Building off of Olof's work, can we extend his work and study the biology of learning anatomy and potentially implement the findings within courses? Sarah ran with and ultimately extended this idea for her PhD. This slide summarizes Sarah's work. As indicated in this slide, we first wanted to determine if we could apply the concept of reinforcement-based learning in anatomy and replicate behaviorally Olof's work. That was Sarah's first piece that was published in 2016. Sarah then followed up with determining if we could use this paradigm to track the neural and behavioral learning curves of novices able to learn the location of 12 cranial nerves. And I'll give you a brief overview of her findings in a few slides. That's her second piece that was published in 2018. Sarah and Heather weren't satisfied. Anatomy is taught in 3 dimensions. However, preliminary teaching and image projection is typically in 2 dimensions. Sarah posed the question, can we identify the neural correlates of learning in 2D, 3D, and in an interleaved fashion and determine which presentation would facilitate better memory recall and allow participants to transfer learning between 2 and 3D environments? And that is Sarah's final paper that was published in 2019. Here are the results of Sarah's first paper using reinforcement-based learning principles for anatomy. She demonstrated that she can take novices and have them go from 50% accuracy, guessing, to 90% accuracy, categorizing 10 neuroanatomical structures in approximately 25 to 30 minutes, using a reinforcement-based learning task, much like what was shown as the exemplar with the blobs in the few slides ago. In our next experiment, Sarah built the same paradigm to track the neural and behavioral learning curves and extend the research in 2 ways. First, she wanted to test retention, so she had participants sit the reinforcement-based learning task twice, at baseline and 1 week later. Second, she was interested in transfer, so she had participants learn anatomical structures by diagram and then presented them with photos. She had 23 participants participate in a series of learning modules to assess the utility of reinforcement-based learning as measured by both accuracy and neural profiles. Her findings were Behaviorally, they were similar to what was found in her first study: increased accuracy and decreased time for trial completion between early and late phases of the experiment. Neurally, reward positivity decreased during module 1 of the study from early to late, indicating learning occurs, and this coincided with the behavioral data. Early reward positivity is in the black line. Late reward positivity is in the red line in the diagram on the right-hand side. The diminished reward positivity amplitude to positive feedback is maintained on a knowledge retention exercise, and that's demonstrated by module 2. And reward positivity amplitude remains diminished as learners successfully transferred their learning to a new context, and that was also within module 2, 1 week later with photos. This suggests that reward positivity was maintained, so participants were able to self-regulate as participants transfer knowledge from a diagram to a photo of a real brain. The basic premise for this work is that through this recognition-based task, fundamental recognition of information could be used, say, before class time to recognize anatomical structures. This does not mean that participants gathered or understood foundational knowledge, just that they recognized what term was associated with what particular structure. If this can be performed before class, then teaching can be made more tailored and thus maximize teaching time, or utilize just-in-time teaching methods within curricula in different courses as you move forward. And this is the really exciting piece that Sarah had done. And so in Sarah's final piece, she created a paradigm using technology to compare learning of 2D versus 3D anatomical structures in masked, blocked, and interleaved situations, measuring behavioral and neural learning and retention. Sarah first administered a pretest to show no difference in 2D and 3D knowledge of the anatomical structures. She then created 3 groups of approximately 20 participants and had them undergo a reinforcement-based learning task, either 2D, 3D, or in a combination of 2D and 3D anatomical models followed by a post-test. She then had them come back 30 days later and complete an identical task that included 2D and 3D models to assess retention and transfer. The hypotheses of this experiment were that 1, that reward positivity amplitude would decrease as ability to internally evaluate the correctness of responses improved. 2, learning from 3D models would be more efficient than 2D and result in diminished reward positivity amplitude. And 3, learning in an interleaved approach will improve long-term retention and result in lower reward positivity amplitude during retention test. Needless to say, this work generated a ton of interesting data. So what I'm focusing on are the reward positivity components. Presented are the amplitudes of the 2D, which are blue, 3D orange, and 2D, 3D, which are green. In module 1 on the left-hand side, you can see that there's no difference in amplitude in the first block, but there is separation between the 3 conditions, and it shows that that in the interleaved condition, participants are relying less on external feedback and it is retained over time, as is shown in the module 2 results. Sarah concluded that based on the neurological results, interleaved learning incorporating both 2D and 3D models provided an advantage in learning, retention, and transfer. Interestingly, she showed that the behavioral data did not have the same sensitivity for differences in learning with and without stereopsis, and she's pursuing that line of research as well. So at roughly the same time as Sarah's work was going on, Chad Williams in Olaf's lab was interested in whether we can apply reward learning and reward positivity in a clinical categorization task. So working with clinicians, we were interested in whether these paradigms could be used to train undergraduate students to identify patterns to select correct diagnoses. Specifically, Chad had 26 students who had no medical training, learned to categorize 5 liver diseases. Basic premise again being that through this recognition-based task, fundamental recognition of information could be used before class time to recognize patterns for diagnosis. This does not mean that participants gathered or understood foundational knowledge, and why those patterns led to diagnosis, just that they recognized what patterns were associated with diagnosis. The theory being that if recognition or the cognitive framework is somehow created, instruction can then provide the necessary details to the framework developed so that students can recognize clinical presentations and create diagnosis or differential diagnosis. Chad used a variation on the reinforcement-based learning paradigm by showing cards with different clinical values, and these values were presented in different locations on the card over the course of the experiment. The participants were then presented with 4 choices, given feedback, and based on those choices. Chad again showed that increased accuracy and decreased response times between early and late phases of the experiment indicated an improvement in categorizing liver disease types, suggesting that this paradigm and the use of simple feedback can be used to identify key diagnostic classification features. Again, with Chad's brain activity results, we saw that the reward positivity amplitude decreased over time, suggesting that reward positivity amplitude is sensitive to learning throughout this task. These findings suggested that participants were relying initially on feedback to confirm their decisions. After participants learned to classify diagnostic disease types, their expectations would be the same as their outcomes. Amplitude of the prediction error would be reduced and the trend would lower. As a result of all of these studies at the University of Calgary, the undergraduate medical education group and academic technologies team have brought to life the use of this within the UME curriculum. So they are now using cards and reinforcement-based learning tasks in order to supplement course content across the curriculum based upon the findings presented. The website I've just presented here, and if you're interested in this fascinating area and the work that's being done, I would suggest either contact me later or please look up Mike Paget at U of C and take a look at the fascinating work that is being done currently. Continuing to build off this work, we kept coming back to Olav's 2009 paper. There were differences in those that were classified as high achievers and those that were classified as low achievers. This begged the question, is there a difference in novices and experts in the reward positivity as they undertook this reinforcement-based task? Working with colleagues Amy Warren, a veterinarian that has her board certification in both clinical and anatomical pathology, as well as a PhD in medical education research focusing on perceptual expertise in eye tracking, and with Sarah Anderson, we recruited vet pathologists within Alberta and 27 undergraduate students. We gave these participants the same reinforcement task with vet pathologies. We combined eye tracking and EEG to determine if we can combine these data sources to verify if both visual perception and neural profile converge as participants, specifically novices, gain proficiencies in recognizing clinical pathologies. Shown here on the left-hand side is Nia, at the time an undergraduate student working on this project, and she's wearing both the EEG and eye tracking equipment. What is shown on the right is unpublished preliminary data that we're incredibly excited about. So there's a slight change on the y-axis. In this case, positive is actually up. It shows that the expert neural profile is relatively unchanged across the trajectory of the experiment. So they weren't influenced or relying on feedback of the case presentations, whereas the novices show a change in trajectory of this reward positivity over time, which over the course of the experiment begin to match the expert profile, which is predicted by the neural reinforcement theories. What the previous few examples show are the application of studied phenomena in applied health education context. As I indicated at the beginning, this work creates more questions than it has answers. For instance, what is the flexibility and adaptability of these frameworks? There are questions regarding information integration and retention, even questions regarding potential rigidity and foreclosure on clinical presentations given the scenarios and informations are presented in only one way. Ultimately, there are many pieces of work that continue to be explored. While these preliminary experiments provide evidence of neural learning curves that could be used for perceptual expertise, the research I presented is lab-based, where students have to come to us to participate. Given student workload and demands on student health and health professions education is high, during the non-pandemic time, it was difficult to get students, not necessarily undergraduate students, but health profession students, to participate in these studies, let alone veterinary and medical clinicians. So, moving forward, we have been piloting the use of mobile EEG technology to capture performance that is going to be more in situ. So Kergolson et al. Published a validation study comparing brain activity data collected with the Muse. So the Muse is in the right-hand upper corner. The paper itself is in the left, and compared the data collected from the Muse to high-density arrays and showed that this mobile EEG data are of comparable research-grade quality. We then followed up this work with collecting brain activity from 1,000 participants in malls, schools, and different settings to assess brain activity and cognitive fatigue. This was just published in Frontiers entitled Using Muse. We've actually started to implement that into some different studies. And so there's 2 other proof of concept studies that were currently being written up to demonstrate the use of these devices in order to assess cognitive fatigue in more real-world settings. One looking at brain activity of practitioners, both pre and post call, and that's that picture in the lower right. As well, we have one tracking ourselves and a research team that went to the High Seas Mars Space Analog, where we spent a week living in a simulated Mars space habitat. However, is the use of these devices worth the effort? What does it contribute to the betterment of teaching, learning, decision-making, and ultimately patient outcomes? We are still a long way off understanding where, how best, and when to use these technologies to facilitate learning. Some may recall Brian Hodges' talk and his reference to the Wall Street Journal clip documenting the use of neural headbands in classrooms in China. Is that what we're attempting to accomplish? From a scientific perspective, obviously, I'm curious to collect those kinds of data. However, as EEG data are messy and there is work that needs to be done to optimize the use of these methods in health professions education. In closing, well, I showed you how we are operationalizing a specific learning task, adapting a known research paradigm, and looking at one ERP component. There are numerous other questions to be asked, domains to be explored, and technologies to be assessed. In my lab, we have a few other projects in the learning, decision-making, and motor skills domains. Most of these studies were begun pre-COVID. An unintended positive consequence of the pandemic for us is that it made us pause, reflect on our research questions, our research paradigms, the quality of the data that we are collecting, and the analytical methods that are being used. This process in itself would be a larger discussion to have with people. So while I'm incredibly excited to continue the work in this field with this amazing team of professionals and a long lost friend who I get to do research with, I want to leave you with some of the challenges that have been discussed in the literature regarding neuroeducation research and that we constantly consider and debate in my lab. First of all, ecological validity. Do we force participants to act in artificial ways in these environments and in the lab? What is the level of complexity of tasks that we can actually ask these participants to undertake within our lab-based environments? Do we have or can we mimic the level of complexity that is encountered in the classroom during a clinical skill or during a clinical encounter? Second, reductive bias. So learning, expertise, decision-making is reduced to electrical activity and blood flow. What about the context of environment? What about the effect of stress, peers, supervisors? So this is actually pushing us or moving us to an area called social neurosciences. We have a preliminary experiment going on in my lab to look at the effect of cooperative and competitive learning environments while scanning 2 people at one time. Implications for educational practice. Daniel Ansari and others have written about the caution of rushing too quickly in this area. The suggestions being that results shouldn't be directly applied, but implemented and translated in a gradual and iterative process. Indeed, much work at the applied level And really, we need to determine if work can optimize our understanding of these educational practices by using these data. To bring it back full circle to Brault Fawzy's comments regarding the areas of neuroeducation, can we examine the biological process that support or constrain learning? Should we explore the neural correlates of academic skill? And can we or should we investigate how teaching practice or educational interventions impact brain function and structure? Finally, ethical and philosophical challenges. What are the ethical implications of collecting and using these data for learning, teaching, and decision making? Should we be collecting these data from our students and potentially our raters and teachers, much like biometric data, and especially within this era of big data? So in this talk, I wanted to provide a specific example of how we move from a basic sciences concept into applied neuroeducation research to the possibility of incorporating those findings into classroom settings or learning settings, and really moving from blobs, basic sciences, all the way to liver pathologies. Where I first thought this research would lead to more answers, I find myself asking more questions and realizing it requires a team of research expertise to continue to work in this field and a lot of time to do this work. I do look forward to this challenge and continuing to do research in this area. So with that, what I'd like to do is say thank you and just show you the members of the Phenol lab, as well as all the investigators that I get to work with. If you would like to contact myself or any members of our lab, please go to our website or just email me directly. And what I'd like to do is conclude. Thank you, and I look forward to your questions.
Thank you, Kent. Thank you, Kent, for a fascinating talk. I think you've generated a series of questions already in our Slido. I'll give people maybe a minute to get some questions up there. Let me just make sure I'm coordinated. So let's see, the first question up here is: the early work you presented talked about high learners and low learners, with low learners not decreasing the reward reaction, suggesting they did not learn the material. This factor didn't get carried through the later discussion. In education, how can we be able to identify or assist low learners?
I think that's an excellent question. And in Olaf's original blob papers, if you go back and take a look at it, he actually had them in 10 blocks with 120 trials per block. In our subsequent papers, and we mentioned this actually in Sarah Anderson's work, we had fewer number of blocks and fewer numbers of trials. And I think we weren't really able to distinguish between those within the more practical applied settings. And so that's really what we wanted to accomplish with that late liver pathology piece and looking at differences between novices and experts as a way of seeing where there would be differences and whether or not we could identify that neural profile. So I agree, that's a great question and one that can be definitely be further explored within this area.
And I guess building on that, the next question, and you brought it up on your last discussion as well, is the issue of ethics around this particular idea of low learners. So have you seen, or do you intend to study how these ERP signals shift depending on whether you're studying, oops, it moved, sorry. Is the idea that eventually instructors could use the neural data to identify who is learning the perceptual content and develop support? Could this also be used to eliminate poor learners? And then the person wrote sticky ethics.
Whoa, this is incredibly sticky ethics. And again, like I said, I am neurosciences adjacent, but there's also a really interesting literature around the philosophy of neuroeducation and the potential impact it could have. So I think it could, much like multiple assessment methods allow us to have a better picture of an individual, I think this could potentially provide a glimpse or another piece of the puzzle that could be used with other data in order to address that question. So I'm kind of sidestepping the question, but I think it is an area of real interest that a lot of work can potentially be done in.
Thank you. Have you seen or do you intend to study how these ERP signals shift depending on whether you're studying acquisition, retention, or transfer of knowledge? How about reacquisition of skills and knowledge? It's a double-barreled question.
And a great question, and like I mentioned before, well, where I thought with our research paradigms we were considering a question, what I'm finding as a result of going through this is it's asking more questions, much like what is posed here. And it's really starting to challenge us to figure out whether or not our research paradigms are what we actually are intending to capture. And so really part of this work, a colleague of mine explained it to me, being an experimentalist, is really finding that appropriate research paradigm in order to start to tease these questions apart. So these are further questions that we're interested in, that we're intending to look at, and that we would like to study.
Thank you. I have a follow-up question, but maybe I'll move to this one first. Thanks for your interesting talk. It strikes me that there's a wide range of content in HP curriculum, for example, from diagnostic reasoning to ethics. Do you have a sense that these approaches are best for some areas of content, but might create different problems if applied to other areas of HP?
I think that, again, that's a great question, and really thinking about what domains should we or could we be using this in is an important question. I don't think it's a one size will fit all. I think we have to be very cognizant of where, how best, and when to use these technologies with the respective questions. As I mentioned before, we have done some work in the psychomotor domain, taking a look at laparoscopic surgical skills, and I think that's another area that's really interesting to take a look at. The ethics questions would be fascinating, and I think in order to move forward within this field, if people are interested, it really takes a confluence of research expertise as well as looking at those different literatures that would allow us to inform how best to move forward in our applied field.
Thank you. The last question on the Slido is, I wonder about the ERP response of people who belong to racialized and other traditionally excluded groups.
That, and again, so that's a female male differences, racialized group differences, all of those components. And this is one question that Olav and I keep coming back to is we need a baseline. We need to understand baseline before we move forward in order to address questions such as this. So that's why I say these are preliminary data that we're presenting. They're really, while there's hypotheses, they're exploratory in nature, and there's these pieces associated, just like what this question poses, that really have to be determined before we think about meaningfully about implementing. And so the other piece that I'm interested in is really where, how best, and when, as I've mentioned before, to use these types of technologies. And we have to be willing to say maybe we should not be using them moving forward.
Thank you. Excellent talk, thank you. After the COVID pause and reevaluation phase, what were your main takeaway points for what to pause on and what you feel is the best forward direction for your work ahead?
This is a whole talk in and of itself. Where do you want to begin? Our research paradigms, and it may not be appropriate for what we're trying to accomplish. The data that we're collecting, are we analyzing it properly is really a big piece. And this really gets to the idea that given our questions, given the theories that we're bringing to bear, if we use different questions and different lenses to look at those data, we potentially may come up with different answers. So it's really interesting to think about it. And maybe what I'll do is give a concrete example. My background is educational measurement and assessment, and I'm actually interested in whether or not we could use this in order to better our understanding of assessment methodologies. So we have a pilot project right now taking a look at multiple choice questions versus script concordance test questions that are matched with novices and experts, a 2x2 design. I thought that would be a fairly easy design to implement, to record data on. What we're finding is those preliminary data are just so complex. I don't know if the actual experimental paradigm is correct. But what we tried to do with that experimental paradigm is make it as most lifelike as possible. But we're finding are the data really messy. So right now we're having fascinating conversations about how best to look at those data, what type of analytical method should we be using. And we're finding we're doing that with that experiment, with other experiments. Like I mentioned, we have a hyperscanning, so scanning two people at one time in order to look at the social interaction that occurs and really discussing what that means and what we could find. So those are the really interesting, fascinating, muddy questions that we're getting to work on. And hopefully well, I don't want to say answers, we'll have more questions about moving forward.
Yeah, I had a question about the collaborative learning piece and how that might fit in with what you're thinking about. I wonder if you can elaborate a bit or hypothesize a bit on how that would look if you were to study collaborative learning interventions.
Oh, this is really interesting. So we've again built this, and this is an Amy grant that we received just to do some pilot work, take a look at team-based learning. So we have an individual from psychology, organizational psychology, that has a team-based background and well published. What we've tried to do is put people into a collaborative situation and see whether or not they would agree or disagree on certain presentations associated with it. And I don't have a picture, but it's actually a really interesting setup of the experimental design. And what we're trying to figure out is this idea of interbrain synchrony. So are they in sync as they move forward, or aren't they? And so we're first trying to figure out whether or not this collaborative work could potentially identify neural correlates that we could use, and then put them into the second piece, different contexts, cooperative environments, competitive environments, different stressors are provided to them, and see how those interbrain synchronies, if you will, would potentially or could potentially change. Then the third piece, if we ever can get there, and if we can ever bring people back into the lab in order to do those experiments, really we thought, wouldn't it be interesting to see whether or not if we could actually use a teaching intervention and see whether or not that intervention would affect how people would interact. That is our dream. That's where we're hoping to get to with that type of work. But I'm more than willing to have conversations, discussions, debates about the opportunities associated with that. There's a larger literature that's coming out in terms of social neurosciences, and some hyperscanning events that have actually used the portable learning devices that have some preliminary pieces of information, but there's just so much to explore to see whether or not we can actually identify A, neural signatures, B, what would happen in competitive or cooperative environments, and see the effects of different teaching interventions on those interactions. I hope that answered your question, Maria. Is that?
Yeah, no, yeah, that's very helpful, and I think it aligns with the next question that's in the Slido with what you're describing. So they said perhaps a less ethically sticky way to use these approaches could be more in terms of exploring the use of these technologies and enhancing learning processes and supporting learning rather than using them judgmentally, which I think is what you just described.
Which I completely agree with. I think this is meant to be. So I look at it as an educationer, as an educationalist, as being supportive of the learning process, and wouldn't it be interesting in order to be able to provide another lens. And this is really why I got into this, is as a measurement person, I always joke that measurement error kept me up at night. Because we're assuming learning has occurred as a result of a behavioral score. Wouldn't it be interesting to use another variable, in this case, a neural component, in order to help facilitate with learning processes and to support learning as moving forward? And I think you'll see that with more of Sarah Anderson's work as she moves forward.
So I don't see more questions in the Slido, so I'll take this as an opportunity to ask some questions that I've been thinking about as you've been talking. And maybe if that generates more questions, that'd be great. People can continue to use the Slido. But I'm wondering how this work intersects with known successful educational interventions like productive struggle or productive failure, where the pattern of the learning might look a little bit different in terms of what the participants are experiencing. So struggle, I don't know how it would intersect with reward potential and how that would look. And I wonder if you've thought a lot about that.
a great question. I haven't necessarily seen that done. Again, what I'm finding is even in the last 7 years, I'm still drinking from a fire hose where reading work such as and opening up from work such as Olav's work as well as other neuroscientists' work and taking a look at more of their basic sciences paradigm and thinking about how we can potentially apply it. Are there pieces that are potentially out there that are asked? Yes. And so what I'm finding is I feel like I'm doing a second or third PhD all over again because I'm learning a whole new literature and trying to apply that literature within our respective discipline. And I think it would be interesting to see whether or not, and this gets back to a comment I made before, just what those baselines are for those respective individuals and to see whether or not we could then move them forward. Because the interesting thing, and just to get to Olav's paper where there was a difference between high and low learners, wouldn't it be interesting in that case to see whether or not an intervention would have helped facilitate them in order to get to a level of accuracy, but B, that deflection and reward positivity to show that they've actually attained a level of understanding of what those, in that case, blobs were to be. So as you can well imagine, there's a lot of potential for doing good work, but it's also a lot of potential of thinking about how to do those experiments sequentially so we build the evidence in order to help them with learning processes and really to support learning.
Thanks. That's very helpful. And it's got me thinking about a number of different things now. We have another question from the Slido. Could these tools be used to assess learning with other technology like collaborative training in virtual reality?
I'll talk to Sarah Anderson. So the 2D versus 3D work that she did, she had an in-video glasses that she was able to flicker between 2D and 3D as people were learning. So she's really interested in VR work and continuing to use that in her work in terms of learning and anatomy. And what the individual here is suggesting is even taking it an extra step and taking a look at it from a collaborative perspective. And rightly or wrongly, I think that people are going to start to use the more mobile EEG technologies. The real question is, what is the signal that we can tease out from that noise? And there's a lot of noise. And so really thinking meaningfully about what we're trying to collect, how we're trying to collect, and importantly, to get back to a number of the questions that were asked, is what is that impact on teaching and learning and education?
Great, thank you. I wonder if in the last few minutes, you've alluded to this throughout your talk, but ask you to step back a little bit and reflect on working at such an interesting intersection, but also one that has required you at this stage of your career to learn a whole other set of things. I wonder if that's something you can reflect on. Oh, and now we got, of course, more questions, but.
So I'm finding I wish I were a lot younger. I'm finding I wish I would have stumbled into this area earlier. What I'm finding, and a lot of people find, I think, within health professions education, is we work at an interesting intersection between a number of different people, disciplines, methods, methodologies, and this is just one exemplar. To be honest with you, I would love just to step back and immerse myself in all of this and to be able to play. And my background is actually quantitative. I love modeling data. I love playing with data. And unfortunately, I haven't been able to spend as much time as I want to playing with these kinds of data so that I have an inherent understanding of those pieces. But what's been really enjoyable is bringing people together from different disciplines and having them actually have the conversations. Interestingly, so one of my former PhD students, Alison Brand, introduced me to a term called the hermeneutic wager, which is basically you as an individual, what's your line in the sand when you go into a debate? And so what I'm finding between the neuroscientists and the educationalists and the psychologists is that we're all asking questions and go, but you can't do this in this kind of a context of neuroscience to say. And I'm saying, well, in an applied world, this is the type and this is the messiness of the area that we're playing in. And so by having those conversations, and I'd love to be able to publish this, but these are just discussions, we are, I think, moving forward and we have a better appreciation of each other's field. And ultimately, the idea is, can we use these in order to look at another variable, in order to measure more variance, if you will, so that we have a better appreciation of how people learn and make decisions. And like I said, eventually, potentially even patient outcomes, regardless of the health professions education component and discipline.
Yeah, no, that's great. I think we can all learn a lot from looking at this type of collaboration across disciplines.
Hmm.
I'll go back to the Slido, which is going to pull you into a different direction again. So when dependent on commercially available products like Muse, what are the limitations versus if you were using research-specific prototypes?
Yeah, that's a great that's a great question. So technologies change, technologies morph. Trying to so do you want to be on the leading edge? Do you want to be on the tail edge, or should you be somewhere in the middle? And who's, if you will, in terms of the technologies, that's another area in terms of development and use. And so again, it would be nice to be funded with an infinite amount of money to be able to create and not be reliant on technologies that are necessarily developed elsewhere. But what we're finding is that by partnering with them, we can then use them and implement them within studies, and especially with the Muse. I don't know if people know the Muse story. It's interesting. It has been developed for meditation, and it's commercially available.
No.
And Olaf has been able to look at and develop it. Not, and there are other products similar to it that are also out there, and I think you're going to see even more products like it out there. And so, really understanding and validating, and in our lab and also in Olaf's labs, we're trying to take it from more higher density arrays, create the paradigm, collect the data, and then try it with these lower density arrays and see whether or not we have the same type of data quality, and then take it out into the more applied setting. And that's really how we're addressing that type of a question. But it's fascinating. And we're actually, I've got another individual in my lab who's a biomedical engineer who's actually taking a look at measuring different types of data quality and brain activity, and it's again it's another area that I have to do more reading in and an understanding of.
And maybe I'll jump to the last question here. Curious how you find the uptake of neuroimaging educational research in the clinical community and readiness of HP leadership to re-examine their traditional approaches in light of this new paradigm.
Yeah, and this is so practicality of this work and the applicability of this work. So I'm worried about, the wow factor. I'm hoping that we are generating good science that provides evidence that will allow us to make some potentially critical and good decisions in terms of affecting education. So it's interesting. Some people are ready for it. Some people are saying this is a great idea. Some people are mention it and look at it and go, I can't see the connection between what you're doing and that ultimate piece. And this gets back to that context piece. If we stick a person in a magnet and see that there's differences in terms of brain activity, does that actually translate, or could that translate into different educational practices? I've got ideas for more, and again, what we're talking about here are cross-sectional studies. I've got ideas for longitudinal studies in order to create those baselines, but those are going to be hopefully rolled out in the near future.
So I think, well, there may be time for this one last question. I think these are two, it's actually one. So as per Maria, the concept of desirable difficulties and the broad idea of the performance learning paradox would suggest that making learning difficult, challenging in the moment can be beneficial in the long term. Mm-hmm. That might suggest that the reward positivity could be reduced in the short term, but would be improved in the long term. How does this connect to your current set of studies and findings? Links to the previous points about retention and transfer.
That's a great question, and it gets to this idea of the longitudinality and how much we actually can retain. And so this gets to some of Sarah's work. The first piece was just retention over a week. The second piece was retention over 30 days. What we're interested in is actually measuring individuals with more portable, higher density arrays over time as they go through their trajectory within a health professions education program. And so it would be interesting. I'm hoping maybe in 5 or 10 years time, if I'm still doing what I'm still doing this, if we can still do this work, I'll be able to hopefully address that question more meaningfully.
Sounds great. We'll look forward to it. And I think that's a great place to stop. So thank you to everyone for joining us today. And thank you, Dr. Hecker, for sharing your time and your expertise. I know that you've generated a lot of questions and ideas for me, and I'm sure the same for our audience. And a reminder to everybody to please fill out the evaluation forms when you have a chance. Thank you all.